The goal of my lab is to understand the biology of Alzheimer's disease by performing deep molecular profiling of human samples. And by doing this, we think that we are going to identify novel genes and variants, create new molecular biomarkers, and identify druggable targets. So what we are doing is we are generating genetic data, transcriptomics, epigenomics, methylation, proteomics, and metabolomics from human brain samples, as well as CSF and plasma. And we are identifying genetic variants associated with these molecular traits that we are integrating with new approaches like Mendelian randomization, colocalization, or fusion approaches in order to identify not only associations, but causal proteins or causal analytes. And this is very powerful approach compared with previous methods in which you just perform association analysis, and we never knew if one analyte was causal, part of the causal pathway, or just a secondary event. And we're using these new approaches to really identify these causal events. And by doing this, we are also learning how the proteins and the molecules interact between them, because sometimes we see that similar genetic variants modify multiple proteins, and that is putting the proteins on the same pathway. Once we identify these causal proteins, we also can analyze where those are potentially druggable, whether there is already an FDA compound that targets those proteins, and we can use those for drug repositioning. So in any case, a brief summary is we are really trying to do this deep molecular profiling of human samples to go and understand the biology of the disease.